Hello guys, we're going to start with our bacterial genetics. In this chapter or in this lecture, we'll be basically talking about DNA structure and the DNA replication. Just to give you a brief idea, in a prokaryotic cell or in a bacterial cell, we basically don't have a well-defined nucleus. So there's a region which is ideally irregular shape region which is present over there called as a nucleoid, where your chromosomes are ideally present. Now, your DNA or your deoxyribonucleic acid is the principal molecule which is responsible for the storage of genetic information. Your chromosome is ideally the single largest DNA molecule in a cell that contains the information necessary for the cell's survival itself. Now, any segment of DNA that is capable of encoding its own replication in a cell is ideally termed as a replicon. So, if you see that your chromosome is the single largest replicon which is present in the cell. Now, plasmids are small extrachromosomal DNA entities which are present over there. They have the ability to replicate on its own. They do not require the chromosome for any help in that. They are basically non-chromosomal replicons. Now, some plasmids have certain extra genes within them which confer special characteristics on a particular bacterial cell. Now, phenotype and genotype. Phenotypic characteristics are expressed by a given character in the environment. They'll be externally visible characters. Genotypic characters are basically the genes which encode those characters. They are a collection of the genes. They remain unchanged. Now, a gene that does not receive its phenotypic expression is ideally termed as a recessor. The gene which overrides the effect is ideally termed as a dominant gene. So, the best example when you're talking about Mendel's law, you have a dwarf and a tall plant. So, the tall is the dominant character, the dwarf is the recessive character. Now, going on to the structure of your genetic material, in a bacterial cell, you'll be having DNA. DNA is converted into an RNA. So, we'll first talk about what is a DNA structure. So, your DNA is your deoxyribonucleic acid. Your RNA is your ribose nucleic acid. Your basic building blocks which are present over there are your nucleotides. You have a phosphate group, a pentose sugar, and a nitrogenous base. So, nucleotides and nucleosides. So, when you have a nitrogenous base which is attached to a pentose sugar, it's ideally called as a nucleoside. A nucleotide is a nitrogenous base with a pentose sugar and a phosphate group attached to it. So, you have a nucleotide and a nucleoside. Now, in your DNA, you have 4 nitrogenous bases. You have your adenine, guanine, cytosine, and your thymine. In your RNA, you have adenine, guanine, cytosine. The thymine is ideally replaced by uracil. Now, your adenines and guanines are your purines. Cytosines, thymine, and uracil are your pyrimidines. Now, the structure is ideally of a double helix. You have a chain of nucleotides which are present over there, okay, which are ideally anti-parallel to each other. Now, there's going to be a base pairing to this double-stranded helix is ideally held together by base pairings which are done between adenine and thymine. There are 2 hydrogen bonds which are placed, and guanine and cytosine, where we're having 3 hydrogen bonds. So, ideally, it is seen that guanine and cytosine, the pairing between GC, that is guanine and cytosine, is much stronger in compared to AT because you have 2 hydrogen bonds over here, and whereas in your GC you have 3 hydrogen bonds. So the complementary base pair is always between AT, GC, and in the case of your RNA, your adenine will basically pairing with your uracil. Okay. If you go on to the DNA structure, you have a double helix which is ideally formed. Okay. So, the phosphate group, so let's consider this as your sugar molecule, that is your deoxyribose, okay. In your deoxyribose, on your 5' end, you have a phosphate group, okay. Whereas on the 3' end, the next nucleotide is basically added, okay. On the first part, the first carbon atom, you have your nitrogenous base which is ideally added. Now, this entire nucleotide is joined to the next one via the 3' end. Okay, so you have a phosphodiester linkage which is ideally formed between the third carbon atom, that is the OH end, and the phosphate group from the next nucleotide, which is at the 5' end. Okay. So this phosphodiester linkage is ideally present in your DNA structure. So since the two strands are anti-parallel, on one end, one strand will be having the 5' end which is exposed. On the same side of the other strand, you'll be having your 3' end which will be exposed. So they're basically anti-parallel to each other, okay, and the base pairing will be between them, okay. Now keep in mind, your purines will always pair with your pyrimidines, never a purine-purine, never a pyrimidine-pyrimidine, okay. Now this is your double helix which is ideally formed. It's a spiral coil-like structure. Now it's a right-handed coil which is ideally formed. The pitch of the helix is approximately 3.4 nanometers. So the distance between two consecutive base pairs is 0.34 nanometers. Okay, and in per turn you have 10 nucleotides which are ideally present. Okay? So this is your DNA double helix which is ideally formed. Now going on to the next slide, we're going to start with your bacterial replication. Okay, so bacterial DNA replication. Now before we start with that, there are certain enzymes which ideally play an important role during the replication process. So we'll just go through them one by one. Now you have the first enzyme which is DNA polymerase 1. Now it has an exonuclease activity which removes RNA primer and it replaces with a newly synthesized DNA. Exonuclease activity means it helps in removal of your nucleotides. Okay. Your DNA polymerase 3 is basically the main enzyme which goes on adding nucleotides in the 5' to the 3' direction. Then you have your helicase enzyme which opens the DNA helix by breaking the hydrogen bonds. Then you have your ligases which ideally seals the gaps between the Okazaki fragments. That is on your lagging strand to create one entire continuous strand. Primase is an enzyme which produces or creates or synthesizes a small RNA primer which is ideally required for the replication to start. Your primase is nothing but an RNA polymerase as such. Now you have single-stranded binding proteins which will bind to the single-stranded DNA to prevent the hydrogen bonding between the DNA strands to take place again and thus preventing the reforming of your double-stranded structure. You have a sliding clamp which will basically hold the DNA polymerase in place when the nucleotides are being added. Okay. You have a topoisomerase 2 which is also called as a DNA gyrase. It tends to relax the supercoiled chromosome to make the DNA more accessible for the replication to ideally begin. It also helps in relieving the stress on the DNA when the unwinding takes place because of the extra supercoiling actually which takes place over there. Okay. Now, you have a NAS enzyme that is your topoisomerase 4 which introduces a single-stranded break into your chromosome which will release the tension as such which is there. Okay, we'll go into the diagram and we'll get to know that more better. So, we're going on to your DNA replication as such. In your bacterial cell, you have a circular DNA structure. So, there's a point of origin called as origin of replication. It is approximately 245 base pairs long and it is ideally rich in adenine thymine sequences, okay, AD sequences. Now, this origin of replication is where the hydrogen bonds will ideally break and the replication will start from that particular region, resulting in a formation of a bubble. So, you have a replication fork, ideally you have two replication forks which are ideally formed, which proceed in both directions of that particular bubble. So the bubble tends to go on increasing till you have the termination of the replication taking place and you'll be having two daughter DNA strands which are ideally created. Now this model is ideally called as a theta model because you have a theta structure which is ideally formed in the middle. So I'll just go into telling you what happens at the replication fork. So this is the next slide. Now if you see this bubble which is over there, you have the leading strand which moves towards the fork and the lagging strand which ideally tends to move away from the fork. Okay. Now I told you the DNA replication ideally has to have or basically goes from a 5' to a 3' position. Okay. So on the leading strand you can ideally have the 5' to 3' direction going on. Okay. Now what happens is that your DNA polymerase 3, which is ideally required for the replication to take place, cannot ideally go on adding new nucleotides just in random way. Okay, it has to have a 3-OH end for it to go on adding nucleotides. Now, the 3-OH end is ideally not available at that particular point. So at that moment, your primase will come and do the work. So your primase will create small sections, okay, approximately 10 to 15 nucleotides long, okay, and it will basically provide a 3-OH end to the for the DNA polymerases to add a next nucleotide on, okay. So on the leading strand, you just have one region or one place where your kinase will work, and the DNA polymerases will go on adding the nucleotides which are ideally required. Okay, I forgot to tell you that you have your helicase enzyme over here, which will basically help in unzipping the double helix. You have your single-stranded proteins, which will bind over there, which will ideally keep the DNA stable, so it does not go and bind up again or create a double-stranded structure again. Okay. Now, on lagging strand, ideally what happens is that the replication fork is in the strand or the parental strand is in the opposite direction. Okay. So now in this case, ideally what happens, small segments are ideally created for the replication to ideally take place. Okay. So small primers are ideally added onto your lagging strand, and then your DNA polymerase ideally synthesizes off. This is because the parental strand itself is in the opposite direction. So the entire thing ideally cannot take place properly. Okay. So you have small primers which are added by your primase enzyme, and then these segments are basically extended off by using your DNA polymerase. Any gaps which are remaining, so these are small sections of DNA which are ideally created. So these small sections are ideally called as Okazaki fragments. Now these small DNA fragments are again joined up by your DNA ligase enzyme to create one long stretch of DNA. Okay, keep in mind your DNA strand is synthesized towards the fork and your lagging strand is synthesized away from the fork. Now, there's another thing. What happens is that these small RNA primers have to be removed off because they are ideally made up of RNA. So, remember I told you about an enzyme that is your DNA polymerase 1 enzyme, which has an exonuclease activity. This comes into play over here. So, your DNA polymerase 1 will eat up the entire RNA primers which are ideally created, and that will be ideally replaced by your DNA nucleotides. So, that will be one entire long stretch of DNA. Okay, so just an overview of the entire replication process. It's a circular, in a bacterial chromosome, you have a circular model. Okay, so a DNA bubble is ideally formed over there. Okay, now the bubble tends to grow in both directions. Okay, so a replication fork is created in both the sides. You have the blue DNA or the blue lines, which is the new DNA, the black lines, which is the old DNA. So your DNA is ideally synthesized in the 5' to 3' direction. You have a DNA helicase enzyme which unzips the DNA. Okay. Your parental DNA strand is used as a template. You have a topoisomerase enzyme which tends to prevent the extra stress on the DNA which is still wound around each other. Okay. So small nicks are ideally created to relieve the stress out, and those nicks are again sealed off by your ligases enzyme. You have your DNA leading strand, which is going to be a continuous strand, which is from 5' to 3'. Your primase, which is nothing but an RNA polymerase, will make small primer. Your polymerases, that is DNA polymerases 3, will basically act on it, go on adding your new nucleotides to the 3' OH end, thus creating a complementary strand towards the parental strand. Now, on the opposite strand, that is your lagging strand, you will be having a non-continuous synthesis of your DNA from the 5' to 3' direction. Okay. Again, small primers are made along the entire stretch using your primase enzyme. your DNA polymerases 3 will go on extending that particular section of DNA by adding your nucleotides to it. Your DNA polymerases 1, showing its exonuclease activity, will eat up all the RNA primase part or a primer part, replacing those strands with your DNA nucleotides. And the Okazaki fragments, which are ideally created on the lagging strand, are all sealed up using your DNA ligases. Okay, so this is all about your DNA structure and your replication. Thank you.